Alright, welcome back, fellow scientists. So today we're going to talk about a process called transcription. Now, just to review where we've been, we found out that there was a transforming factor. We found out that DNA was that transforming factor. We found the structure of DNA, and then we looked at one of the two things that DNA has to do. DNA has to be easily copied. So we just got done talking about DNA replication and how that works with DNA helicase and DNA polymerase. Now the second thing that DNA has to do is we have to be able to take the information in DNA and we have to be able to put that to work. Right? There has to be some way that the information in DNA controls the cells or tells the cells what to do. So that process of taking the information in DNA and putting it into work, that's actually a two-step process. It's called transcription is the first step, which we're going to talk about today, and then translation is the next step, which we're going to talk about in the next video. So let's go ahead and let's write down a definition for transcription. That's what we're going to talk about today. So transcription. Transcription is the process of taking the information in DNA and copying it into RNA, copying the information into RNA. Copying it into RNA. So if we had to summarize the basic process of transcription, all we're doing is we're taking the information in DNA and we are transcribing it. It's the same language, it's nucleic acids, it's just a different form of the language. We're transcribing that information into RNA. Now, please don't think that we're changing DNA into RNA, because we're not. The DNA stays the same, as we'll see, but we're just taking the information in DNA and we're changing forms, right? So before we get into the actual process of transcription, I think it would be useful to look at the differences between DNA and RNA. So over here in this column we'll have DNA, and over here in this column we will have RNA. So the first difference, and this is where the difference in the name comes in, DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid, right? So what does that look like? Well, DNA, deoxyribonucleic acid, it has a deoxy, it's missing an oxygen in its sugar. So its sugar, remember, is a pentagon, and all of these angles right here represent carbons. And then there's a base that comes off here, and then there's another carbon that's right here, and then we have phosphate groups off of there, right? So in DNA, we're going to see a hydrogen coming off of this number 2 carbon right here. In RNA, again, it's the same pentagon. That's a horrible pentagon. You guys can do better in your notes. And then base coming off there, and then a carbon, and then phosphate group. But in RNA, it's going to have an extra oxygen because DNA, deoxy, right, it's missing an oxygen. RNA is going to have an OH, a hydroxyl group, coming off of this number 2 carbon right there. So that's the first difference. Next difference is that DNA is almost always in a double helix. There are some exceptions, but that's beyond the scope of this course, so don't worry about it. DNA is always in a double helix. RNA is most often single-stranded. It's not a double helix. Most often single-stranded. DNA, for our purposes, we're going to say that there's one type of DNA. There's a couple more, but again, that's beyond the scope of this class. And for RNA, for our purposes, we're going to say that there's 3 types. And again, there are more, but that's beyond the scope of this class, so we'll just stick with these 3. The 3 types are mRNA, tRNA, and rRNA. Now, of course, these front letters stand for something. They mean something. mRNA stands for messenger RNA. tRNA stands for transfer RNA. And rRNA stands for ribosomal RNA. You can hear the word ribosome in there. Obviously, it's associated with the ribosome. All right, now, DNA, as we already know, has 4 bases. So it has the bases A, C, T, G. RNA also has 4 bases. 3 of them are the same as DNA, and 1 of them is different. So the bases that RNA has, it has A, C, U, and G. The U is the base uracil, and in RNA it takes the place of T. So no longer in RNA does A pair with T, but A pairs with U. C still pairs with G, just like that. And then we could also say that DNA lives in the nucleus. That's where its home is. So DNA lives in the nucleus. Yeesh, it's horrible. In the nucleus. And RNA, it's made in the nucleus, but it does its job. It spends most of its life in the cytoplasm associated with the ribosome. So it's made in the nucleus. Made in the nucleus, but it lives in the cytoplasm. And usually it's associated with the ribosome. Lives in the cytoplasm, we'll say next to or by. Next to or by a ribosome. Alright, so those are the differences. So now that we know the differences between DNA and RNA, now we can look at the actual process of transcription. Process of transcription. Transcription, in case you can't read scribble. I apologize. So, as always, we start off with a piece of DNA. So here we go. Here's our DNA. We're going to go like this, just like we did with DNA replication. Kind of like that. And like that. And just like with DNA replication, the DNA is in a double helix all the way down, but I'm leaving space here so that I can write letters. We can write A, G, T, C, G, T, C, G, T, A, T, 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 A, T, T. If you want to, pause it and remember, practice our base pairing rules. So check yourself. T, C, A, G, C, A, T, A, like that. Okay, so that's all it is. That's what we start with. Now during, again, we're going to have a little fork or a little bubble. So we're going to make this kind of bigger like this, and then this is going to be a little bit bigger like that. Alright, now there's only one enzyme that's involved in transcription, and that is RNA polymerase. So RNA polymerase does it all. It unzips the DNA, it adds RNA nucleotides, adds RNA nucleotides to the growing strand of RNA, and then it rezips the DNA. Right, and so we're going to have RNA polymerase kind of travel this way. Okay, now we need to go back and put in our bases. So A, G, T, C, G, T, A, T, T on this side, and then T, G, A, G, C, A on this side. Alright, let's see, we'll use blue for RNA. So then, and this is where some people can get a little bit messed up. This is where it gets a little bit tricky. Because we have different bases, because we don't have a T, because we have a U, our RNA is not going to be this exact same sequence of DNA, this complementary base pair, right? So T usually pairs with A, so we're going to have A nucleotide right here. Again, T usually pairs with A. Now A, in DNA, A usually pairs with T, but RNA doesn't have a T, it has a U. So instead now, this A, it doesn't pair with T, but it pairs with U, right? And then here we have TA, and then of course we still have that, and we still have our G. T, but pairs with A, and then we have a C, and then A normally pairs with T, but again there's no T in RNA, so there is a U. So then we have this little strand of RNA right there that's made by RNA polymerase. And RNA polymerase just keeps on going down, and it unzips and it adds nucleotides, and then it rezips, and then after the end product of transcription. So after, we're going to have our piece of DNA just like it was before. We're going to have our same letters just like we did before, so A, G, T, C, G, T, A, T, T, and then of course G, C, A, G, C, A, T, A, like that. But then we're also going to have our RNA. We're going to have like that, and we're going to have U, C, A, G, C, A, U, A, just like that. So that is our mRNA, and that's going to go to the nucleus, or sorry, to the cytoplasm rather. And then this DNA, notice that's the exact same thing that we had before. So the DNA stays in the nucleus, it lives in its home, and it's ready to be transcribed again, or just to kind of sit there and wait until the cell actually needs it. So that's the process of transcription. All we're doing is we're taking the information in DNA and we're changing it, we're changing forms into RNA. Next up, we'll talk about the process of translation, which is the second part of, again, putting the information in DNA to work, using that to control the cell, using that to make workers.